now we're ready to tackle photosynthesis, the process by which carbon is fixed by plants, algae, and photosynthetic bacteria, and the process by which sunlight energy is converted to the food that supplies the energy for all life on Earth. So if we look at the overall reaction for photosynthesis, you notice that we take carbon dioxide and we have water as well. You know that plants require carbon dioxide and we need to water plants in order for them to thrive, to function, to photosynthesize. And out of this we produce in the process of photosynthesis carbohydrates, which are the ultimate food source for all organisms, and we have water and oxygen is released. So plants breathe in carbon dioxide and breathe out, if you will, oxygen, the opposite of what occurs in respiration. And if you were to cancel out, say, 6 of these waters from each side, so we put 6 here and we cancel out these H2Os over here, then what you see is that this reaction is essentially the reverse of the process of respiration. So here we have photosynthesis and here we have respiration. This is essentially the reverse of that. So let's examine how it is that this biochemistry is run. And it's a wonderful system of interacting proteins that are able to harvest the energy that is in light. So we need to study light. And as we pointed out here, oxygen is generated, and we call that type of photosynthesis oxygenic photosynthesis because oxygen is being generated. And we have cyanobacteria that do this, and 7 groups of algae, and of course all land plants. So we have the biochemistry of this system is divided into 2 sets of reactions. The light-dependent reactions, which depend on the energy coming from the sun in the form of light, and then that can be used to make ATP and NADPH, which is another electron carrier that will provide energy for the dark. These carbon fixation reactions are known as the dark reactions. They're not really carried out in the dark. They can be, but they are light independent reactions. So whereas the capturing of sunlight and the production of ATP and NADPH, it depends on light. The light independent reactions, or the carbon fixation reactions, take the ATP and the NADPH generated in the light dependent reactions, and they take that energy and they use it to fix the carbon in carbon dioxide into carbohydrates. So we will study how that occurs. And just schematically here, what we have is the harvesting of light energy by photosystems. And photosystems are large assemblages of macromolecules, of proteins, with accessories, pigment molecules embedded in them. And these pigment molecules serve as antennae to gather light energy and funnel that energy into what we call the reaction center, or the oxygenic reaction center. And at that reaction center, we will elevate the energy of electrons that will be passed to other electron carriers. And in so doing, we will generate ATP and NADPH. And that is, we generate ATP here by phosphorylating ADP, and we generate NADPH by reducing NADP+. And then the NADPH and the ATP generated in the light-dependent reactions by the electron transfers generated ultimately by these photosystems, that will be used in what we call the light-independent reactions in the Calvin cycle to take carbon dioxide from the air and produce organic molecules, fixing that carbon in organic molecules. So this is just a schematic, and the only other thing I want to point out right now is that water is used in the light-dependent reactions. And in fact, water becomes oxidized to oxygen and donates electrons to the photosystems, and those electrons replace electrons that are elevated to higher energy and passed on to other electron carriers, as we will see. And this all occurs in the chloroplasts, and it occurs in the thylakoid membranes that are the inner structures, if you'll remember, of mitochondria. and in the grana, which are stacks of those thylakoid membranes. And remembering that the stroma is the fluid outside of the thylakoid. So here we have going from a plant cell here to looking at chloroplast structure. And here we have the outer membrane of the chloroplast, the inner membrane of the chloroplast, and then here are the thylakoids, which are these flattened sacs, membranous flattened sacs. And they are stacked into what we call grana. And then outside the thylakoid stacks, outside the grana, is what we call the stroma of the chloroplast. That would be the fluid, semi-fluid substance contained within the inner membrane of the chloroplast, but outside the thylakoids. And here's a blow-up of that. Here's an electron micrograph of a chloroplast, and you can see these stacked thylakoids, these grana. So that all this is taking place in the chloroplasts, and the membrane of the thylakoids is where the photosystems are located. And they are the ones that are going to initially harvest the light energy. So because I want to save time for experiments later on in genetics, I'm going to not spend a lot of time on the history of the discovery of photosynthesis, but it basically came down to understanding that plants can produce oxygen, and this oxygen was able to sustain life that was not otherwise sustainable, and that was derived. Those discoveries were made quite early in the 1500s and 1700s, and it was in the 1930s that a chemical formula for photosynthesis was generally worked out. So it was, this is it, carbon plus H2 something plus light energy could yield carbohydrates plus more water plus 2 of the something that was ever here. If this was oxygen, if A was oxygen, then we would generate oxygen. That is, H2A is the electron donor. So, and Van Niel also, in the 30s, identified oxygen as being released from photosynthesis definitively. And this was confirmed. As I said, I'm not going to go over these experiments. You can read about them in your book if you are so motivated. Please do. So let's define the light harvesting pigments and explain how the antennal system funnels light energy. We know that a photon is a particle of light. Light has wave properties, but it also has particle properties. And so photons act as discrete bundles of energy. And it is those discrete bundles that are harvested by the pigment antennae of photosynthetic organisms. So the energy content of a photon is inversely proportional to the wavelength of the light. The shorter the wavelength of light, the more energy it has. The longer wavelength of light, the less energy it has. So think of waves in the ocean. Short, choppy waves have a lot of energy. Long, slow waves have less energy. And the photoelectric effect is the ability of light to remove an electron from a molecule. So a particle of light, a photon, has a photoelectric effect on molecules. It can remove an electron from a molecule when a photon is impinging on a particular molecule. That can remove an electron and excite it to an energy level that allows it to then be transferred to derive energy. So this photoelectric effect, as I've said, this photoelectric effect occurs when photons transfer energy to electrons. And if we look at the wavelengths of light that are involved here in photosynthesis, we see that it's only a tiny part of the electromagnetic spectrum. So at the very low end of the electromagnetic spectrum, we have very short wavelength light. We have gamma rays. Oh, I'm sorry, we don't even have light there. We have gamma rays, X-rays, and ultraviolet light. But in terms of visible light, that doesn't appear until close to 1,000 nanometer of wavelength. So this is a very narrow part of the entire electromagnetic spectrum. Visible light occupies only a tiny part of that. And if we blow up the visible light spectrum and go from 400 nanometers to 740 nanometers, we see that at the shorter wavelengths of visible light, we have the blues, and moving into the greens, yellows, oranges, reds, and then kind of far red. And right at the border of visible light, we have the junction between far red visible light and the infrared. Some organisms can see infrared or UV. We can't. This is our visible light spectrum. So the light energy harvested by the photosynthetic pigments in plant cells actually, as we will see, composes different parts of this visible light spectrum. Only a tiny part of the electromagnetic spectrum is harvested by plants. So let's look at the pigments that absorb the visible light in chloroplasts. And if we look at those pigments, we see that each has a characteristic absorption spectrum, which is the range of light, the range of visible light in terms of wavelength that can be absorbed and be used to excite electrons. So if we look at, for example, the pigments found in chloroplasts, we have carotenoids, we have chlorophyll A and chlorophyll B, and we see that each of these pigment molecules, and there are many of them in a particular photosystem, as we'll see, have characteristic wavelengths at which they absorb light most intensely. And you'll notice that in the kind of a pale green to yellow range, these pigments absorb very poorly. And so if you were to deny plants light in these wavelengths, or both these wavelengths, there would be very little photosynthesis if all they had available to them was just yellow light. But in fact, they have the whole range of visible light coming to them from the sun. So chlorophyll A is the primary pigment in plants and cyanobacteria as well. And let's go back here. Chlorophyll A, if we look at its absorption spectrum, we see that it absorbs highly in the blue range, and secondarily it has a second absorption peak out here in the red range as well. So let's look at these pigments. If we look at chlorophyll A, we see that it absorbs both violet blue, as I've said, and red light, whereas chlorophyll B is a secondary pigment, and it absorbs wavelengths that chlorophyll A does not. In fact, if you look at their absorption spectra, you see that chlorophyll B absorbs light, it has a peak absorption well outside of the peak absorption of chlorophyll A at the lower range of its absorption. Its action spectrum, we would say, where it absorbs light. So these secondary pigments then with absorption peaks different from chlorophyll A, the accessory pigments, which are carotenoids as well as chlorophyll B, increase the range of light that is efficiently used in photosynthesis, because chlorophyll A is a pigment which is the primary pigment for capturing light energy in photosynthesis. But these accessory pigments broaden the range of visible light that can be used to harvest energy. And chlorophyll B, as I've said, is a secondary pigment that absorbs light wavelengths that chlorophyll A does not. And if we look at the structure of pigments, we see that they're made out of porphyrin rings, which are complex rings with alternating double and single bonds. And there is a magnesium ion coordinated by atoms in the center of the porphyrin ring. And these rings are pigments, these photosynthetic pigments, these porphyrin rings are capable of having electrons that are excited by photons of light as the photons impinge on the porphyrin rings. And in so doing, the electrons that are excited are shuttled away from the porphyrin ring of a particular pigment. So here is a chloroplast, and here is a membrane in the thylakoid within a chloroplast. And here we're representing in this blue blob is a photosystem. And we'll talk about the photosystems shortly. But this is a photosystem, and it's a complex of proteins with embedded pigment molecules shown in green in them. So we could have chlorophyll A, chlorophyll B. There are others as well, the carotenoids, and there are even other ones, other pigments as well. And this is the basic structure, though, of a photosynthetic pigment. Here's the porphyrin ring structure. Here is the coordinated magnesium ion, coordinated by nitrogen atoms in the porphyrin ring. And there's a hydrocarbon tail as well. And it is these porphyrin rings then that capture the light energy that will be transduced, as we will see, into chemical energy.